My name is Mila Staub. I'm a sophomore at Franciscan Missionaries of Our Lady University, majoring in nursing. For this presentation, I will be discussing microbial pathogenesis and how it relates to influenza. I will first be discussing the differences between pathogenesis and pathogens, and then I will be moving on to microbes and then microbial pathogenesis. After microbial pathogenesis, I will move into the influenza background and influenza prevention. Finally, I will end off relating microbial pathogenesis and influenza. Jumping right in, this slide will give a better understanding of the differences between pathogenesis and pathogens before going more into depth on microbial pathogenesis. A disease's pathogenesis describes how the disease develops, progresses, and if the disease persists or becomes extinct. Pathogenesis is a product of the interaction between an infected pathogen and the immune system. Pathogens cause infectious diseases. Types of pathogens include fungi, worms, and viruses, as well as the others on this slide. However, most bacteria are not pathogenic. Every type of pathogen has its own way to enter a host and disrupts the host's immune system. Now let's talk about microbes. There are many different types of microbes. Some microbes include bacteria, protozoa, fungi, archaea, algae, viruses, and prions, as well as the other ones listed on this slide. Other than viruses, these microbes can live without being attached to a host cell. Viruses can only live for a brief time without attaching to a host cell. To go more into depth on bacteria, protozoa, and fungi, bacteria have a simple cell structure when compared to the other organisms. They are unicellular organisms with DNA in a single loop. Bacteria can be found all over the earth, from the ocean and soil to the inside of our bodies. Protozoa is a more complex organism than bacteria. Protozoa is a single-cell eukaryotic organism. They reproduce asexually, and a key indicator that is different from bacteria is the presence of a nucleus. Protozoa can be found in a wide range of places, such as soil and fresh water. Fungi is a group of eukaryotic organisms. Fungi includes yeast, mushrooms, and mold. The difference of fungi can range in size and be either unicellular or multicellular. Of these fungi, yeast is unicellular and mold is multicellular. Viruses are different. Viruses are responsible for many diseases that occur in plants, animals, and even humans. Viruses are the most simple when compared to other organisms. They have no cells or organelles. Viruses cannot live long outside of the host cell because without the host cell, they cannot replicate. Many scientists disagree with the idea that viruses are considered alive because of this reason. In microbial pathogenesis, microbes or microbial components cause the infection in a host after interacting with both the host and the pathogens. When it comes to the human microbiome, these microbes are not harmful. They actually help with many everyday processes that the body undergoes. Some of these processes include digestion, regulation of metabolic pathways, and they also help to regulate the body's immune defenses. However, when these healthy microbes travel to a part of the body where they are unwanted, it can cause infections. Pathogenicity is the ability of a microbe to cause a disease or damage the host. Pathogenicity is based on virulence factors. Virulence factors allow the bacteria to latch onto or enter the host to cause disease. Most of the virulence factors that allow the bacteria to cause disease are quorum-sensing genes. These genes sense the size of population and lead the individual bacteria to act as a population. This is a major part of pathogenicity and survival for bacteria. Now to transfer over to influenza. Influenza is a viral infection that takes place in the respiratory system, particularly the nose, throat, and lungs. Most of the time, influenza can resolve on its own, but in some cases, it can be deadly. Before vaccines and the annual influenza seasons, there were lethal strains of influenza that would emerge. In the last century, there have been 3 global pandemics for these lethal strands. The worst pandemic occurred in 1918. It was called the Spanish flu. This strain of influenza killed an estimated 50 million people. Animals can also contract influenza. The same animals can give the virus to humans. These types of influenza that affect mainly animals include avian influenza, which affects mainly birds, and swine influenza, which affects mainly pigs. These types of influenza are seen in the 2009 influenza pandemic, which is a mix of avian, swine, and human gene strands. It is important to know the symptoms of influenza. There is a wide range of symptoms that may align with other viruses. To be protected from influenza, there are vaccines. The vaccines are not 100% effective, but they can lessen the symptoms and later complications. There are also rapid tests that can determine if someone has influenza. These rapid tests allow you to stay isolated if you are proven positive for influenza, and this can lessen the spread of the virus. The rapid test takes about 30 minutes to provide results. Finally, I will discuss the relation between microbial pathogenesis and influenza. Influenza viruses are human and animal pathogens. As stated before, pathogenesis is the product of an infected pathogen infecting the immune system, and pathogenicity is dependent on the virulence factors. In the 1918 pandemic, influenza's virulence factors were unique, and they had an age-specific death toll. There was a spike in deaths of certain age groups during this pandemic. Since influenza is transmitted through the respiratory tract, they mostly replicate in the epithelial cells lining it. Influenza kills the epithelial cells, and when the epithelial cells are infected, they release cells to attract inflammatory cells. These inflammatory cells just cause more death through the epithelial cells. To recap or sum this up, the inflammatory cells are brought in to help, but they actually cause the death to the epithelial cells. This means that the pathogenesis of severe influenza is not caused by the viral effects, but it is caused by the overcorrection of the host's inflammatory responses. To end this presentation, let's recap on what we learned. Infected pathogens enter a host and infiltrate the immune system through the ability of pathogenesis. If a microbe is harmful, it is considered a pathogen. Microbial pathogenesis is the ability for these microbes to enter the host and cause an infection. The pathogenesis of influenza is when the virus enters and kills the epithelial cells through respiratory transmission. In relation together, microbial pathogenesis is when microbes cause disease, and in influenza, pathogenesis is driven from the influenza virus. The references listed are the references that helped me to create this presentation.